Hello everyone. So today we are going to start about the topic that is central dogma. So central dogma is the process in which the double-stranded DNA known as replication is converted into RNA, that is transcription, and then the RNA convert into protein, that is known as translation. So the process of replication, transcription and translation together known as central dogma, which is very essential and crucial for all living beings. So replication occurs in both prokaryotes as well as eukaryotes. So let's study the general review of all the prokaryotes and eukaryotes. Replication occurs in prokaryotes has three major steps, that is initiation, elongation and termination. Initiation consists of DNA A protein, DNA B protein, DNA C protein, which binds to 9-mer sequence. That's why melting of 13-mer sequence starts. Second step is elongation, where helicase, SSV and DNA polymerase 3 is responsible, and termination is done by gyrase, also known as topoisomerase. Then comes to replication in eukaryotes. Replication in eukaryotes also has three major steps, that is initiation, elongation and termination. Initiation includes CDC6 protein, CDT1 protein and MCM helicase. And for the activation of this, CMG complex is needed. Then elongation is done by polymerase epsilon for leading strand, known for continuous strand, and polymerase delta and alpha for lagging strand. Termination, where two replication fork meets. This is all about the replication in prokaryotes and eukaryotes. Then comes to transcription. Transcription is the process in which the DNA is converted into RNA. And for transcription, there are several steps like initiation, elongation, termination. So let's discuss about transcription in prokaryotes. Transcription in prokaryotes, initiation contains a sigma factor and holoenzyme. Elongation has core enzyme and gyrase, and termination is done by row dependent and row independent. Row dependent is done by row protein, row independent is done by hairpin loop structure. Now comes to transcription and in eukaryotes. Transcription in eukaryotes has three major steps: initiation, elongation, termination. Initiation has TBP protein, TAF protein, transcription factor D protein, transcription factor A protein, transcription factor BFE protein along with DNA polymerase along with RNA polymerase 2 for initiation. For elongation, topoisomerase 1, 2 and polymerase 2 is responsible. And for termination in eukaryotes, RAT1 protein is needed. Then comes to translation in prokaryotes. The conversion of the RNA into protein is known as translation, and translation has three major steps: initiation, elongation, termination. Initiation has initiation factor 1, initiation factor 2, initiation factor 3. Initiation factor 1, 2 and 3 responsible for 30s, 50s subunit, together form 70s subunits. For elongation, transcription factor TU, transcription factor TS, elongation factor G is responsible. For termination, release factor 1, release factor 2, release factor 3 is needed. Then for translation in eukaryotes, initiation, elongation, termination is needed. Eukaryotic elongation factor 2, 1, 1A, 3, 5, 2B, 4G, 5B needed for 40S initiation complex. Together form 40s and 60As form 80S subunit. Then for elongation, eukaryotic elongation factor 1a, eukaryotic elongation factor 1b, eukaryotic elongation factor 2 is formed. And for termination, eukaryotic release factor 1, eukaryotic release factor 2 is needed. So it's all about the central dogma. Thank you.